Welcome back. So today we're going to be doing genetic growth of prokaryotic cells. Yay! Okay. So first you need to know is the main way prokaryotic cells divide is through binary fission. Okay. So pretty much it's a type of asexual reproduction where cells divide into two identical daughter cells. Okay. Meaning we could have one big cell that turns into that. Okay. It is very quickly and it's very effective. And again, it's very common among bacteria, but also different type of prokaryotic cells. Okay. Awesome. So prokaryotic cells, prokaryotes can exchange material through three different ways, right? We have conjugation, transformation, and transduction, which are all different forms of genetic recombination. So let's break down each process. Okay. So first, let's look at transformation. So prokaryotic can take DNA from their environment, okay, either from lysed cells or released DNA, okay, meaning that like we know lysed means a cell that has been like when a cell lyses, it pretty much means it blows up, okay? So what's happening here is the cell, let's say it blows up, leaves a bunch of stuff all over the place, and then the prokaryotic bacteria can take that DNA. And then this DNA can then be integrated into the recipient cell genome and leading to genetic changes. Okay, so the second step is, not step, sorry, these aren't steps, but the second type is what we call conjugation. So this is direct transfer. I remember I drew it on the last video, it looked pretty bad, but this is pretty much direct transfer. So you could look a lot like, again, draw my ugly drawing again. It looks even worse this time. It's like anyways, let's say this is a cell and that's a cell. They're going to be passing DNA. Sorry, terrible drawing. But yeah, so that's that. Again, it really is just moving. Yeah, exactly. Known as a mating bridge. You could imagine it as a bridge connecting one place to another. Literally like people walking across the bridge. So this involves the transfer of plasmids. So plasmids are small circular DNA molecules. And then the third type of genetic recombination is transduction. So usually a virus, in particular, we call it a bacteriophage, transfers bacterium from one to another. So a few things these phages can do, they can actually accidentally, keyword accidentally, package bacterial DNA during its replication cycle and then transfer that DNA to a new host. And the last thing, it was so weird. Kaplan just like had this in their very last page, totally out of. Anyways, I just don't think it's very, like, it doesn't really pair up to what we're doing. But transposons are able to remove and add themselves into a genome. And the reason why I think this is confusing is because this chapter is about prokaryotic cells, but actually, this can actually also happen in eukaryotic cells. So this isn't prokaryotic specific. Awesome. Hopefully that video was helpful. Next, we're going to get into viruses. And this is the last section for chapter 1. So after that video, we'll get into chapter 2. See ya.